Assalamu alaikum warahmatullah. Today our lecture about the muscle tissue, and our objectives for this lecture will be to identify the structure of the muscle tissue, the structure of the three types of muscles, and to describe the structural and functional differences between the three types of muscles. The contents of this lecture will be skeletal muscle, cardiac muscle, and smooth muscle. The three types of muscle tissue: skeletal muscle, cardiac muscle, and smooth muscle. There are certain terminologies we have to know while studying the muscle tissue, like sarcoplasm, which means the cytoplasm of muscle cells, and the sarcoplasmic reticulum, it is the smooth endoplasmic reticulum in the muscle cells. And the third term is the sarcolemma, which is the muscle cell membrane and its external lamina. So we have sarcoplasm, is the cytoplasm of the muscle cell, sarcoplasmic reticulum, the smooth endoplasmic reticulum, and the sarcolemma, which is the muscle cell membrane and its external lamina. The skeletal muscle fibers are long, cylindrical, multinucleated cells, and the diameter of the skeletal muscle fiber is about 10 to 100 micrometer. As we see in this image on the right side, this is the skeletal muscle fiber, and we notice that it's long, cylindrical, and multinucleated. We have multiple nuclei in each skeletal muscle fiber. And we notice the striations. The elongated nuclei of the skeletal muscle are located peripherally, just under the sarcolemma, which is the muscle cell membrane. This is the muscle fiber or the skeletal muscle cell, and we notice these are the nuclei, and they are located peripherally على الأطراف, just under the sarcolemma. In the skeletal muscle there are, or in every type of muscle, there are thin layers of connective tissue that surround and organize the contractile fibers. These layers of connective tissue are arranged بطريقة معينة to surround and organize the contractile fibers of the skeletal muscle and the other types of muscle also, but it's well seen and well presented in the skeletal muscle. The epimysium and the perimysium, and the third one is the endomysium. What is the epimysium? The outer layer, the external sheath of dense connective tissue that surrounds the entire muscle. ده اللي بيحيط بالمصل كلها من برا اسمه epimysium. From the epimysium, septa of this tissue extend inward. بتمتد septa إلى داخل المصل, carrying the larger nerves, blood vessels, and the lymphatics of the muscle. Okay, these septa carrying larger nerves, blood vessels, and the lymphatics of the muscle. So this is the external sheath, is the epimysium. Let's see in the image how it looks like. If this is a bone, we have this bone, and then we have the tendon. From the tendon we have the muscle. This is the skeletal muscle. Okay, the skeletal muscle, the outer layer, dense connective tissue layer, is the epimysium. As we see here, this is the reflected layer. This reflected part is the epimysium. Okay, surrounding the whole muscle with its spondylus and uh, containing vessels and nerves from the outer side, the epimysium. The perimysium is a thin connective tissue layer that immediately surrounds each bundle of muscle fibers. عندنا المصل مكونة من muscle bundles or fascicles، وأي muscle bundle is composed of muscle fibers. each muscle bundle is surrounded by perimysium. So البيريمايزيوم موجود surrounding each bundle of muscle fibers or each muscle fascicle. Let's see in the image. As we see, this is cross section through the muscle. We notice this and this rounded bundles of muscle fibers. These are called fascicles. This one is muscle fascicle, and this muscle fascicle or bundle. These fascicles are surrounded by the perimysium. We took one of them out like this. This is a muscle fascicle, or as we notice, muscle fascicle. This one is a muscle fascicle surrounded by perimysium. Okay, so the perimysium is surrounding each muscle fascicle or muscle bundle. Within the fascicles, the endomysium surrounds the external lamina of the individual muscle fibers. Let's see in the image. This is the fascicle. Within the fascicle, we have many individual muscle fibers. This one of them, one of the muscle fibers, surrounded by the endomysium. Okay, the endomysium surround the external lamina of the individual muscle fiber. Okay, and this individual muscle fiber is a muscle cell, a skeletal muscle cell, containing inside the nuclei and the myofibrils. الدوائر الصغيرة دي هذه these are myofibrils. So the muscle fiber is surrounded by endomysium, the muscle fascicle or bundle surrounded by perimysium, and the whole muscle surrounded by epimysium. And superficial to the epimysium is deep fascia. دي بيدرسكم لها في الجروس anatomy. The deep fascia. احنا concerned بس بالendomysium, perimysium, and epimysium. A longitudinal section the skeletal muscle fibers show cross striations of alternating light and dark bands. يعني في longitudinal section يعني skeletal muscle هنلاحظ للstriations في جزء light جزء dark. The dark bands are called the A bands and the light bands are called the I bands. Let's see in the following images. On the left side we have a longitudinal section of skeletal muscle stained by H&E, hematoxylin and eosine. On the right side we have also a longitudinal section of skeletal muscle stained by Giemsa stain. We notice there is striations, dark bands, light band, dark band, light band, continuously. The dark band is called the A band and the light band لونها pale أو فاتح دي هي ال I band. Okay, and the right side we have the Giemsa stain section, very clear, the striations within the section of the skeletal muscle. And these dark bands are A band, the white bands are I band. In the transmission electron microscope, each I band, here the lighter band, is seen to be bisected by a dark transverse line, the Z disc. مثلا دي الصورة اللي على اليمين، ده صورة هاخدها بالإلكترون مايكروسكوب مايوفيبريل عشان نشوف ال I band وال A band. This dark region is a A band. This lighter region من هنا لهنا is a I band. قال لنا إنه in the middle of the I band is bisected by dark line. This dark transverse line, and this is called the Z disk. Okay, this dark line found in the middle of the I band is Z disk. Okay, M stands for mitochondria. These M rounded structures are mitochondria. And in the middle of the A band is a lighter zone called H zone. هنتكلم عنها في السلايد الجاي. The A band shows the presence of a lighter zone in its center, the H zone. طيب عندنا دي ال A band من هنا لهنا. Okay, لاحظين this is the dark region is called the A band, and this is a lighter region is the I band, and the I band is bisected by transverse line called Z disk. And this inside the A band we have a lighter zone in its center. دي الجزء ده كله, this lighter zone in its center is called the H zone. Okay, and bisecting the H zone line in the middle is called M line. Okay, so in the A band we have a lighter zone in the center called the H zone, and in the middle of the H zone there is a line bisecting the H zone called M line. The repetitive functional subunit of contractile apparatus, the sarcomere, extends from Z disk to Z disk. الجزء من ال Z disk ده لل next Z disk, this من هنا لهنا, this is called sarcomere when contractile apparatus, and this is the functional subunit of the contractile apparatus اللي هي دي بتكون مكررة على طول المصل المايوفيبريل. وبنلقى from disk to disk, from Z disk to Z disk is the sarcomere, which is the functional subunit بتاعنا. تمام. What about the sarcoplasm, cytoplasm? The sarcoplasm has little rough endoplasmic reticulum. It contains primarily long cylindrical filament bundles called the myofibrils. The myofibrils, they are running parallel to the long axis of the fiber. اللي شفناها في ال previous photos of epimysium and perimysium, we noticed the myofibrils. The skeletal muscle also, the A and I banding pattern in sarcomeres is due mainly to the regular arrangement of thick myosin and thin F-actin myofilaments. يعني قبيل ال A band اللي هي darker region and the I band the lighter region is due to what؟ في شنو في الحتتين ديل؟ موجودة ال thick myosin filaments and the thin F-actin myofilaments. arrangement معين بطريقة معينة أدتنا جزء يكون darker، الجزء التاني يكون lighter. هنشوف الارينجمنت ده شكله كيف. Let's see first what is myosin and in structure, the structure of the myosin and the structure of the actin. The thick myosin filaments are 1.6 micrometer long and 15 nanometer wide, and they occupy the A band as the middle region of the sarcomere. تمام؟ يعني عارفين احنا ال A band اللي هي قاعدة في النص لأنه الساركومير from Z disk to Z disk. The A band or darker region in the middle. The myosin is found there in the A band. The thin helical actin filaments. So the actin filaments are thin and helical. Okay, they are each one is 1 micrometer long and 8 nanometer wide, and these actin filaments they run between the thick filaments, and that means between the myosin filaments. Thick filaments we mean myosin filaments. Thin is the actin filaments. In this image we see on the left side the muscle fiber, which is a skeletal muscle cell. Inside this muscle cell we have sarcoplasm containing myofibrils, these round structures are myofibrils. Each single myofibril contains myofilaments, which are these myofilaments are either thick filament, myosin, or thin filament, the actin. The thick filament or the myosin is composed of what? It's the thick in comparison to the actin, thin filament, and each part of it, for example this one, we have this structure within the myosin filament. In this structure we have head and tail. So the myosin molecule is composed of head and tail. In the head we have the actin binding site and the ATP and ATPase binding site. This head is responsible for the sliding along the actin filament during the muscle contraction. In the thin filament or the actin filament we have also it's composed of G-actin, this one molecule is G-actin. This group of molecules is called F-actin, and we have this green one is a tropomyosin, and this blue molecule is a troponin protein. All of these are proteins. In the troponin we have the calcium binding site. Okay, once the calcium is bound to this site, the actin filament will be activated and the myosin binding sites will be open to receive the hits of the myosin to slide on it, and all of this occurs during the muscle contraction. This is the arrangement of the thick filament myosin and the thin filament actin. As we noticed before, we have the A band, which is the darker region, the darker area, and the I band, which is the lighter region. In the I band we have actin filaments that does not overlap with the myosin, only purely actin filaments with the Z disc in the middle. And in the Z disc we have actin binding protein alpha actinin, which binds these actin filaments together. In the A band we have the thick myosin filaments and the overlapping parts of the actin filaments. Okay, this is inside the A band. In the middle of the A band we have a lighter region. This lighter region is composed only of thick myosin filaments. In the middle of the H zone, we have the M line, which also consists of myosin binding proteins. Actin filaments are also tightly associated with two regulatory proteins, as we mentioned in the image, the tropomyosin and the troponin. I bands consist of the portions of the actin filaments that do not overlap the myosin filaments. The A band contains both myosin filaments only and the overlapping portions of actin filaments. The H zone, corresponding to a region with only the rod-like portions of myosin molecule and no thin filaments. The H zone اللي هو الجزء lighter in the middle of the A band نلقى فيه only the part of the myosin molecule and there is no thin filaments. The M line contains a myosin binding protein called myomycin and the enzyme creatine kinase. So the M line contains a myosin binding protein, myomycin, and creatine kinase. In skeletal muscle fibers, the sarcoplasmic reticulum is specialized for calcium sequestration، تخزين وحبس الكالسيوم في الداخل. دي الساركوبلازميك ريتيكولم بتاع السكليتال muscle fiber. بيحصل له release upon certain stimulation by motor innervation, motor impulses from nerves. The sarcolemma is folded into a system of transverse or T-tubules. sarcolemma تكلمنا عنها هي المسل cell membrane يحصل له folding inside the muscle cell into transverse or T-tubules. الترانسفيرس T-tubules دي مهمة جدا في contraction of the skeletal muscle and they form a triad with the terminal cistern of the sarcoplasmic reticulum. The terminal cisterns of the sarcoplasmic reticulum, they contains the calcium. They lie adjacent to the T-tubules and they form triad with the T-tubule and two terminal cistern. And the T-tubules, they surround and encircle all the muscle myofibrils so that when the contraction occurs, becomes strong enough to contract the muscle and move the skeleton. We have finished the skeletal muscle. Now we are moving to discuss the cardiac muscle. The cardiac muscle cells are approximately 15 micrometer in diameter and from 85 to 100 micrometer in length. As we see in this image, this is how it looks like. Muscle cells, cardiac muscle cells, they are branching and communicating. They exhibit a cross-striated banding pattern. Also there is cross-striations. إذا ركزنا نلقى فيه striations very clear on the cardiac muscle. Each cardiac muscle possesses only one or two centrally located nuclei. النيوكليوس بتاعتها شنو؟ موجودة في النص، ممكن تكون واحدة أو اتنين but it is not multinucleated unlike the multinucleated skeletal muscle. The multinucleated skeletal muscle fibers, the nuclei are multiple and located peripherally. And the unique and distinguishing characteristic of the cardiac muscle is the presence of intercalated discs اللي هي بتظهر لنا as dark staining transverse lines that cross the chains of the cardiac cells at irregular intervals. Let's see in the image. In this histological section of cardiac muscle, longitudinal section through the cardiac muscle, we will notice these dark transverse lines, this one, and it's labeled by I, intercalated disc. This transverse line, and here, all these are intercalated discs. N for nucleus, these are the nuclei, nucleus, and nucleus. S for striations, to notice there are some striations in the cardiac muscle. So the unique and characteristic feature for the cardiac muscle is the presence of the intercalated discs. These are the intercalated discs. They cross the chain of the muscle, cardiac muscle cells. So from here to here, this is one cardiac muscle cell, and from here to here, this is another cardiac muscle cell. These intercalated discs represent the interface between the adjacent muscle cells and contain many junctional complexes. أمثلة للjunctional complexes اللي موجودة تحديدا في الانتركاليتد disc هي desmosomes, fascia adherens similar to zonal adherens in the epithelial cells, and the gap junctions. تمام. الانتركاليتد disc في in ultrastructural or under electron microscope, it appears having a longitudinal part and transverse part. In the transverse part we have desmosomes and fascia adherens. In the longitudinal part we have the gap junctions. So the intercalated disc represent the interface اللي هو بيكون البوابة أو ما بين كل خليتين بتاعت cardiac muscle في intercalated disc. and it contains many junctional complexes, ودي مهمة جدا في transmission of the electrical impulses from one cell to another cell. The longitudinally oriented parts of each disc have many gap junctions. زي ما قلنا longitudinal part بتاع intercalated disc فيه gap junctions. gap junctions دي بتعمل ايه؟ provide the ionic continuity between the cells. يكون في ionic continuity between the cells, electrical continuity, and these gap junctions act as electrical synapses, and they allow the cells of the cardiac muscle to act like a sensation as a one unit. sensation يعني one unit. okay. فده بيخلي إنه الخلايا بتاعت الكاردياك muscle to contract and act as one unit like sensation, multinucleated sensation. من يحصل impulse جاي من one cell بتنتقل بسرعة لل other cells و they contract at the same time as one unit. This is called sensation. So allow the cells of the cardiac muscle to act like a sensation. The structure and function of the contractile proteins in the cardiac cells are essentially the same in skeletal muscle. نفسها الcontractile myosin, actin. Again, there are some differences معينة في بعض الstructure. T-tubules مثلا are more numerous and larger in cardiac muscle, and the sarcoplasmic reticulum is less abundant. فالتي-tubules أكتر وأكبر في الكاردياك muscle من السكليتال muscle، والساركوبلازميك ريتيكولم أقل. الحاجة الثانية الثالثة إنه الكاردياك muscle cells they contain more numerous mitochondria، فيها أعداد أكبر من الميتوكوندريا بالمقارنة بالسكليتال muscle. ليه؟ due to the need for continuous aerobic metabolism. إنه الكاردياك muscle cells محتاجة بصورة مستمرة لأيروبيك ميتابوليزم، والأيروبيك ميتابوليزم بتقوم به ميتوكوندريا. طيب خلصنا الكاردياك muscle. Now we are moving to the smooth muscle, and the smooth muscle is also called the visceral muscle. The fibers of the smooth muscle are elongated, tapering, and non-striated cells. شايفين؟ This the smooth muscle smooth muscle cells. The fibers of this smooth muscle, the smooth muscle cells are elongated, tapering, non-striated. There are no striations. زي ما شايفين هي elongated spindle shape. عندنا central part بيكون wider, peripheral part is tapering and narrower. Each smooth muscle fiber is enclosed by a thin basal lamina and a fine network of reticular fibers, the endomysium. زي الارينجمنت بتاع شبيهة بالارينجمنت بتاع السكليتال muscle. إنه أي muscle fiber smooth muscle fiber enclosed surrounded by thin basal lamina و the endomysium. The endomysium اللي هو بيكون fine network of reticular fibers. The length, the smooth muscle fibers, its length ranges from 20 micrometer in the small blood vessels to 500 micrometer in the pregnant uterus. يحصل لها increase in size during pregnancy. Okay. Each cell has a single long nucleus located in the center of the cell. Each smooth muscle cell عندها single long nucleus وقاعدة في السنتر. دي حاجة characteristic برضو للsmooth muscle. This is a histological section to show smooth muscle cells in the wall of small intestine. The wall of small intestine عملوا فيه cross section stained with H and E. الجزء ده the inner circular cells ماشي circularly surrounding the lumen of the small intestine as a tube, and this is the outer longitudinal. في الكروس في اللونجيتودينال بيظهر بالشكل ده. الكروس في الكروس سيكشن في الانر سيركولار يكون بالشكل ده. These arrows indicate the nuclei of the smooth muscle cells. Okay, and so this how it appears under light microscope. All the cells of the smooth muscle are linked by numerous gap junctions, and the mitochondria, polyribosomes, rough endoplasmic reticulum, and the Golgi apparatus are concentrated near the nucleus. The caveolae are short membrane invaginations present on the smooth muscle cell surface. It's called caveolae. So the caveolae are short membrane invaginations present on smooth muscle cell surface. This is instead of T-tubules found in the cardiac and the skeletal muscle. Here there are no T-tubules, there is caveolae in the smooth muscle. The fibers have rudimentary sarcoplasmic reticulum. Also the smooth muscle lack T-tubules, and the caveolae contain several pumps and ion channels, and they may serve to organize the proteins, signaling the calcium release at the myofibrils for during contraction of the muscle. The smooth muscle is specialized for slow, steady contraction and is controlled by a variety of involuntary mechanisms. Smooth muscle under involuntary control. The cardiac muscle also is under involuntary control, and the skeletal muscle under voluntary control. Because the skeletal muscle is under somatic motor innervation control, the smooth muscle and the cardiac muscle are controlled, their contraction is controlled or regulated by autonomic innervation, involuntary mechanisms. The actin and myosin myofilaments in the smooth muscle are organized differently from those of striated muscles. In the smooth muscle cells نلقى البندلز بتاعت ال thin and thick myofilaments الactin and myosin myofilaments crisscross obliquely through the cell يحصل لهم criss crossing oblique in oblique pattern زي ما شايفين in this diagram or in this graph. This is one smooth muscle cell and this is the other one. The narrower part of one cell lies near the wider part of the other cell, and we notice that the thick filament and thin filaments, they are found obliquely here and also here. They are found obliquely through the cell. They don't form striations like no striations like in cardiac and skeletal muscles. And we notice that adjacent cells physically they are coupled at dense bodies. هنتكلم عنها dense bodies next. And the myosin filaments have a less regular arrangement and fewer cross bridges than in striated muscle. الارrangement بتاعها less regular وفي fewer cross bridges than in the striated muscle. ده regarding the myosin filaments. Right. Regarding the actin filaments in the smooth muscle, they lack troponin. ما في troponin. بدلا عن التروبونين, the actin filaments they have instead calmodulin and calcium sensitive myosin light chain kinase MLCK used in the contraction mechanism. الكالمودولين والcalcium sensitive MLCK myosin light chain kinase, they act in the contraction mechanism instead of troponin because there are no troponin. There are no troponin proteins in the actin filaments.